Okay, I'm informed that we are now live and good to go. So good day, everyone, and a very warm welcome to all of you linking in to this webinar. This is a joint contribution by the Asia Pacific Malaria Elimination Network, better known simply as APMEN, as most of you will know by now, and also the Outreach Network for Gene Drive Research. The webinar is intended to complement the global World Mosquito Day event, which was actually yesterday. Just to remind ourselves, APMEN is a network that supports 22 member nations in Asia Pacific to reach their malaria elimination objectives, largely through the 3 technical working groups and also 3 interest groups. While the Outreach Network for Gene Drive Research is a platform to raise awareness and promote responsible development of gene drive technology undertaken by the various member institutions comprising the network. It focuses on informing the public about the potential benefits and challenges of gene drive research, particularly in areas like public health and conservation. We're going to have a bit of a mixed bag of presentations today that may at first glance seem to cover a very broad range of topics. We will learn that what we call malaria is not caused by a single parasite, but by many different parasites, and it is not limited to humans. So we're going to learn about zoonotic non-human primate malaria, avian malaria, and gene drive technology to combat this. And we will learn about the disturbing invasive urban malaria in Africa, and also our growing public health threat, which is dengue. All these divergent topics are part of a common theme, which is mosquito-borne disease, their diverse impacts across global society, either in public health or as conservation threats to biodiversity, and the escalating level of these threats and the need to come up with innovative solutions. We have 4 international experts to set the scene, and then after the presentations, we will have a panel discussion to address some of the questions raised by you, our respected audience. But first, please allow me to mention some housekeeping issues. The entire webinar is being recorded and will be made available for anyone to access via a URL that will be shared in the chat box. Please note that we are not able to take live questions from our audience, but please listen. Please feel free to place questions in the Q&A box, and we will try our level best to respond to as many of your questions as possible during the Q&A discussion. Suggestion here, when you insert a question, I suggest you precede your question with the name of the panelist who you would prefer to answer the question. And for example, Dr. Kimberly, what is and then outrolls your question. Please note that we will only monitor the Q&A box for questions, not the chat box. It just becomes too complicated to do too many things at once. So please put your questions in the Q&A box. One more thing, so you're aware of it. There are several WHO RBM endorsed journalists in the audience. So you may want to consider not inserting any libelous comments in the chat box unless you want to risk having it taken up and published in some global newspaper or media outlet. We will have 4 presentations, all back to back, and the Q&A session will follow afterwards. Our first presenter today is Professor Kimberly Fornas, who will talk to us about the emergence of zoonotic malaria in Asia. Also known as monkey malaria, many people are not aware that the sister species of our human malaria parasites are becoming a significant problem in Southeast Asia. And Kimberly will explain this to us. All will be revealed. Dr. Kimberly Fornas is an associate professor at the Saw Swee Hock School of Public Health, National University of Singapore, where she also co-leads the Climate, Environment and Health program. She is also adjunct professor at the London School of Hygiene and Tropical Medicine and the University of Glasgow. Her work integrates environmental data and machine learning to understand and mitigate the impact of environmental change on infectious disease dynamics, including zoonotic malaria in Southeast Asia. And in addition to all of this, Kimberly is also chair of our APMEN interest group on climate, environmental change and malaria, where she's doing a really astounding work. Thank you for setting aside time for this webinar, Kimberly. Over to you.
Excellent. Thank you so much for the introduction and for organizing this. I am very pleased to be speaking about one of my favorite topics. Can you see my slides okay?
Looks good. Kimberly, please go ahead.
Okay, great. So I will speak a bit today about the emergence of zoonotic malaria in Asia, and particularly thinking about some of the ways in which environmental change has really driven the emergence of this disease. So I think one of the first things that can be quite different for those of us who work on malaria is actually thinking of malaria not as just a human disease, but recognizing this can also be a zoonotic disease. So there's very strong evidence that malaria originated in non-human primates, the common kind of non-zoonotic human malaria species, but we also know that there are Plasmodium species that circulate in different wild non-human primate populations globally. And some of these can spill over into humans and cause human malaria infections. Really, I think the strongest evidence that we have on the public health importance of zoonotic malaria comes on Plasmodium knowlesi, which is a type of malaria found in Southeast Asia. So like all malaria species, this is transmitted by Anopheles mosquitoes. In this case, an Anopheles mosquito typically bites an infected macaque and goes on to bite a person who can then develop malaria with very high rates of severe disease and often fatalities as well if this is untreated. So again, where we've really seen some very strong evidence and a kind of a real push towards surveillance of this has come from Malaysia. So Malaysia has had a highly effective malaria control and elimination program. Malaysia has actually eliminated all non-zoonotic malaria species. So there have been no indigenous cases of falciparum, vivax, and other non-zoonotic species reported since 2018. But what we've seen over the past few decades is we've actually seen this increase in the total number of zoonotic malaria cases reported in people. So there are now thousands of human cases of Plasmodium knowlesi that are reported in people in Malaysia every year. So while we know that Plasmodium knowlesi is an emerging disease, it has all the characteristics of a disease that is increasing, it's not a new disease. So this was first described in the 1930s, and it was shown experimentally that this Plasmodium parasite could infect people. So we knew this could cause human malaria. There was really no evidence that this happened naturally until the 1960s. In the 1960s, an American who was working in Malaysia came down with malaria. He was sent back to Walter Reed Medical Center in the US, where he underwent a rather extensive number of non-typical tests to determine what malaria parasite he was infected with. And this showed that he was in fact naturally infected with Plasmodium knowlesi. Since the 1960s, there was really no evidence that Plasmodium knowlesi was at all a public health issue until 2004. And in 2004, Professor Balbir Singh, along with colleagues in Sarawak and Malaysian Borneo, identified that actually there were a large number of human cases which had been diagnosed as Plasmodium malariae by microscopy. And these were, when used molecular methods, actually showed these were in fact Plasmodium knowlesi. So since 2004, there's been human cases of Plasmodium knowlesi that have been reported across Southeast Asia and parts of South Asia. And so, of course, the more you start to look for something, the more you find. So with improved diagnostics, there's been increased recognition of Plasmodium knowlesi and increasing ability to diagnose this disease. But even for accounting for all of this, we do see there's very distinct spatial heterogeneities in places which have the highest Plasmodium knowlesi risk. So Malaysia reports the highest number of human cases of Plasmodium knowlesi, and most of these are concentrated in Borneo, particularly in the Malaysian state of Sabah. And when we kind of start to think about what may be driving this increase, one of the things that becomes quite apparent is that during this time when we saw this increase in human cases of Plasmodium knowlesi, these areas that are experiencing the highest increases of Plasmodium knowlesi were also reporting some of the highest rates of deforestation globally. So as we've been trying to better understand this, we've been really trying to understand what role environment and deforestation has in increasing human risk of Plasmodium knowlesi. So we really have increasing amount of evidence on this to show the strong connection between human Plasmodium knowlesi risk and land cover changes such as deforestation. So these very early studies, looking at Plasmodium knowlesi clinical cases reporting to hospitals and clinics, many of these cases reported some type of forest exposure or activities in Sabah and Sarawak in Malaysian Borneo. Subsequently, a case control study actually looked at identifying symptomatic cases of clinical Plasmodium knowlesi and comparing these to people who were living in the same villages to identify individual risk factors. And this showed a strong association between increased individual risk of clinical Plasmodium knowlesi and different types of forest and plantation activities. Since then as well, we've looked much more broadly at population scales, and we've been able to show that there's a strong association between deforestation and forest fragmentation, so the presence of these forest edges or these very patchy landscapes with increased risks of exposure and infection to Plasmodium knowlesi across the population. So another question that this always raises is not just where are we finding Plasmodium knowlesi in people, but because we know this is a zoonotic malaria species, where are we actually finding the infected wildlife reservoir? So to start to look at this question, we conducted a meta-analysis looking at all surveys across Southeast Asia that had tested non-human primates for Plasmodium knowlesi using blood samples and molecular methods. So in total, this was over 6,000 non-human primates that were included in these different studies. Most of these were long-tailed macaques, some other macaque species, including pig-tailed macaques, and a few studies that looked at langurs and other non-human primate populations. From this data, as difficult as it is to collect data on wildlife populations and to really think about testing all of these different populations, there's quite wide uncertainty in the prevalence in these different wildlife populations. But what we do see is the areas where we have the highest human risk, particularly in Borneo, correspond to the areas where we have the highest infection rates reported in the wildlife population. Additionally, what we found is there is a very strong association between infection levels in the wildlife population and forest fragmentation. So often we find that macaques or other non-human primate species who are living in cities have quite low infection rates. And similarly, the few studies that looked at macaques in kind of more intact closed primary forest found relatively low infection rates compared to macaques that were living in this quite patchy landscape. We additionally found the highest prevalence in long-tailed macaques, which is, for any of us who live in Southeast Asia, know this is a species we come into contact with quite often. Of course, kind of trying to understand the dynamics of Plasmodium knowlesi or any malaria species really requires better understanding the vector ecology. So there have been a number of different studies that have been conducted across the region looking at the Anopheles leucospirus group, which transmits this parasite. One of the kind of key things that has emerged is of these key vector species, these are primarily outdoor biting. So these are not biting inside houses, but mostly outdoors. And really critically, when we start to think about malaria control, these species are biting in early evening times, so between 6 p.m. and 10 p.m. So if we think about when a person might be inside sleeping under a bed net, this is probably when they're still out doing different activities and much more likely to be exposed to these different mosquito species. We've also shown very strong associations between the different habitats and land use types and the abundance of these mosquito species. So typically we found relatively low abundance in primary forests, so these very old growth intact forests. We find lower but kind of quite patchy abundance in different types of plantation and agriculture. But we really find the highest numbers of Anopheles leucospirus mosquitoes typically in these secondary forests, so often in these highly disturbed forests or near forest edges. Another thing that we've been thinking a lot about is as we see this big increase in cases, does this just mean that people are coming into contact with infected macaques more frequently in areas with these mosquitoes? So we have these three populations that are increasing in kind of contact and proximity. And is it just land use, or are we actually seeing a change in transmission? So are we still seeing that the primary mode of transmission is a mosquito biting a macaque and then going on to bite a person? Or is it possible that a mosquito could bite an infected person and then go on to bite another person? So we do know that this kind of non-zoonotic transmission scenario where a mosquito transmits Plasmodium knowlesi from a person to another person is possible because this has been demonstrated experimentally in laboratory settings. We've done a lot of work with our partners in Malaysia, really trying to better understand some of the evidence around this. So from systematic reviews we've conducted, looking at some of the genetic characteristics, as well as the patterns where we see the spatial and temporal patterns of human cases, all of this is strongly suggestive that there's no widespread non-zoonotic Plasmodium knowlesi transmission. So we don't see the spatial patterns that would be consistent of one person getting a malaria case and then a next person getting a Plasmodium knowlesi case near this first initial case in the time that it takes to pass through a mosquito. So while we think this is quite unlikely, and this is still driven primarily by zoonotic spillover, this doesn't mean this could change in the future. So there's a real need to think about how we collect this type of surveillance data that allows us to actually understand these risks. So the other thing I wanted to highlight is, of course, we think a lot about Plasmodium knowlesi because this is the species where they have the highest evidence of public health burden, but this is not the only zoonotic malaria species which is circulating in Southeast Asia. So we have seen infections with people from cynomolgi, from inui, from other Plasmodium species that can be carried by macaques and different types of wild non-human primates. Additionally, I think it's really worth highlighting that this is not just a problem for Southeast Asia. This is a global challenge. And in particular, we work quite closely with colleagues in Brazil and in other parts of Latin America who are looking at Plasmodium simium and Plasmodium brasilianum, which are circulating in wild non-human primate populations within these areas as well. So this is a real challenge when we start to think about how do we move towards malaria elimination when we have this wildlife reservoir of malaria parasites. So I think all of this together really highlights some of the challenges of zoonotic malaria. We are living in a world where we have unprecedented rates of environmental change. We know our climate is changing very quickly. We also know our landscapes are changing in different ways. And this, of course, is creating new risks. We have an open question that we still don't know the answer to about what is the impact of changing levels of immunity as other malaria species are eliminated. So are we starting to see increased risks of zoonotic malaria because immunity to other species may be decreasing? I think one thing that's really important to think of is actually there are no current control measures which are shown to stop zoonotic malaria transmission. So many of our existing malaria control measures are really thinking about breaking this transmission cycle. So making sure to find and treat cases to prevent them from spreading this to another person. If we think that zoonotic spillover is a primary mode of transmission, we really don't have any good measures that can target the macaque population. So aside from limiting our own exposure to mosquito bites, we don't have a way to really break this transmission cycle. So I think this all really highlights what I think is a very strong need to think about really innovative surveillance and control measures. So how do we move forward and how do we think about how we can use different types of data or design new control measures that can address this threat? So with that, I would like to thank many partners who have been involved in this work over many years now. And thank you very much.
Well, thank you so much, Kimberly. Very, very interesting presentation, very well presented, jam-packed with loads of information, much of which I suspect most of us didn't really fully know about previously. So thank you for sharing those insights with us. We're going to come back to you during Q&A session. But thank you for joining our webinar and making yourself available for sharing this information, Kimberly. So I'm going to move on to our next speaker, who is Professor Fredros Okumu, and he'll be telling us about invasive mosquito species, tackling the spread of Anopheles stevensii mosquitoes on the African continent. Dr. Okumu is Professor of Vector Biology at the School of Biodiversity, One Health and Veterinary Medicine, University of Glasgow. Formerly, he was the director of the Ifakara Health Institute in Tanzania, where he led research with global partners and served on advisory groups, including the WHO Malaria Advisory Group, MPAG, and the Gates Foundation's Malaria Strategic Advisory Panel, MSAP. Fredros holds master's degrees in applied parasitology, geoinformation science, and international health management, and a PhD in infectious diseases from the London School of Hygiene and Tropical Medicine. His research is centered on the biology, control, and elimination of mosquito-borne diseases, notably malaria. So in short, Fredros is a highly accomplished and respected scientist, and it's a pleasure to have you as one of our speakers. Thank you so much, Fredros. I'm going to hand over to you, please.
Thank you very much, Leo, and thanks to all colleagues. As Leo said, my name is Fredros. I am a professor at the University of Glasgow, and for a long time I served as the Director of Science at the Ifakara Health Institute, where most of my mosquito research is still based. Today we will speak about Anopheles stevensii in Africa. This is an invasive vector species that originated from from Asia. It's not indigenous in Africa. It has been detected since several years back. Before we proceed, I just want to highlight that Anopheles stevensii is one of many biological threats facing malaria control and elimination programs in the continent. Others threats that we do have include drug resistance, insecticide resistance. We also have failing diagnostics, and occasionally you end up with a number of epidemics here and there. So all these things converge, and Anopheles stevensii is one of those biological threats. So how long have we had Anopheles stevensii in Africa? There was some indications in early 1960s of detections in Egypt, but those were refuted. So the evidence that we have confirmed now is from the reports in Djibouti, the detections in 2012, which I think were actually published in 2013, but these detections were made in 2012. 7 years later, WHO put out the first alert. This was in 2019. At the moment, that is the distribution, as you see in the map there. I always wonder myself whether Anopheles stevensii is ever going to cross the equator. Downward seems to have some kind of latitudinal restriction zone over there. It seems to be spreading westward. There's been detections in Ghana and Nigeria. But we wait and see. There's a lot of people working on this, and hopefully it doesn't spread any further, but signs are that it will spread. Now, there was a recent publication just a few weeks ago, actually, from colleagues at Harvard that suggested that Anopheles stevensii already occupies 13% of the world, so 13% of the Earth's surface, affecting nearly 40% of global population. They also say that this is going to expand. They anticipate that the area of suitability for Anopheles stevensii is going to reach probably a third of the Earth's surface. Now, obviously, these are trends. They could be mitigated, or they might continue as such, but that is the data, or those are the projections that we have at the moment. How did Anopheles stevensii get into Africa, or how does it spread in Africa? There have been a number of hypotheses. I think the strongest at the moment is that it came in through the sea trade, the trade routes. It came in through the oceans and all that, and that it's spreading by road within the continent. There is also an hypothesis that it might be spreading by high altitude winds. In fact, Professor Tovi Lehman, who works a lot on these high altitude movements, has asked always the question of, okay, so if this thing was coming in through the trade routes, how come it's not in other ports? How come it just happened It seemed to have happened only in the Horn of Africa. So perhaps there are other ways that it might be spreading. Nonetheless, there is clearly some inland translocation, which most likely is mediated by inland transportation roads. Do we have a problem with it? Is it going to end up with a lot more malaria there? Obviously, I mean, when William Tarkin and Steve Lindsay first wrote about this a few years ago, they suggested that this was going to be a big threat, especially in urban areas. And it's particularly important because urban areas in Africa have a lot more people, proportion-wise. And so even if the percentage numbers there are small, often you're dealing with a very large population. So that's a concern for sure. Has this been proven? I think in the very beginning, when the Djibouti case arose, there was this graph that showed malaria cases and went up, and by 2020 it was at the peak. And reports from the French army working in Djibouti did suggest that they did not find any other vector species in the city. They found only Anopheles typhonsi. So that way you could say perhaps that obviously that this upsurge was associated with Anopheles typhonsi. There have been models that suggest that you would have a 50% increase. But perhaps I think the strongest evidence of Anopheles typhonsi contributing significantly to this epidemiological burden has been from Ethiopia, from my friend Tadese and group over there, Fitsum and team over there, who have demonstrated the role of Anopheles typhonsi spreading, in this case, drug-resistant malaria that is also impossible to detect with conventional rapid diagnostics. Beyond this, there is something else, which is what happens if people in urban areas in Africa start to worry about a new disease or malaria in the cities. Often when you have fear like this in large populations, it might divert or interfere with the way countries control malaria. So imagine if in Nairobi or in Dar es Salaam or in Lagos, people start to say, oh, we have Anopheles typhonsi here, it might not be a big threat there, but the amount of resource that will be required to counter it in these large urban cities, when these city dwellers start demanding bed nets, might just mean that we do not have sufficient resources left to deal with the bigger burden in rural areas. The other concern is that Anopheles typhonsi also spreads Plasmodium vivax. So will this lead to expansive spread of vivax, which at the moment is not common in Africa? The biology of the vector, we do know now, obviously, it breeds in a lot of these human-made habitats. It breeds in water containers in urban settings. It survives at extremely very high temperatures, so much higher than traditional Afrotropical malaria vectors. And unfortunately, the populations in Africa appear to be resistant to nearly everything. They're resistant to pyrethroids, organophosphates. They're resistant to a lot of classes. Anopheles gambiae and arabiensis tend to still be susceptible to like organophosphates and so on. But it appears that Anopheles typhonsi is much more hardy vector there. And then, of course, it also spreads Plasmodium falciparum. On the other hand, however, when you look at the propensity of the vector to blood feed on humans appears to be very little. I mean, compared to Anopheles gambiae, for example, which feeds 90% of the time only on humans, Anopheles typhonsi somehow does not have such high human blood indices. So it mostly blood feeds on animals. So that kind of means that it might not be a very good vector, but we wait and see, right? So, and plus, I mean, so you have Anopheles gambiae, Anopheles finescens, arabiensis, and Glucksia, which in Africa are like the big guys. Anopheles typhonsi is coming in, but it's coming into urban areas where you have a lot of human population. So we have to consider all this in that context. Can we control it? At the moment, vector control in Africa relies a lot on insecticidal indoor interventions, ITNs, IRS. Because of the behavior of Anopheles typhonsi, it doesn't appear to be very amenable to this intervention. So we've got to figure out something else. Can we use larval source management? I think there is a big opportunity there to do larval source management. Personally, I think that is really our best chances against Anopheles typhonsi. Not larviciding, but really the management of these habitats. But there are several other interventions that are being developed or tested. And I think some of this should be tried also against Anopheles typhonsi. My friend Dave Jackie Charwood has suggested that we could put spatial repellents in traveling buses or trucks, and maybe things like transporting, which also have mortality impacts, could do a lot of good there. Cities can implement bylaws to regulate water storage, and I think there is a lot of opportunity there to work with the local councils to control Anopheles typhonsi. What do I think? I think that yes, Anopheles stephensi is very important. We did write a paper with Professor Abraham Nzava a few years ago, where we argued that yes, stephensi is a big problem, but addressing it should not necessarily divert our attention from Gambier, Rabiensis, Funestus, all those major guys. Obviously, there's a clamor. There are people who believe, there's been an argument that we should aggressively go after Anopheles stephensi and eliminate it. And I wonder, perhaps this is a good thing, and it's inspired by stories such as Fred Soper's work in Brazil. But I wonder whether, if we can mount such an effort, why don't we mount it against Anopheles arabiensis and Anopheles stephensi? This is not to lessen the impact of stephensi, but it's just to say that our efforts against the vector must be context specific, and it must be integrated with the control of other vector species as well. Surveillance of stephensi is difficult yet very important. We got to know where it is, but we do not know yet how to do it. At the moment, we're not even sure if we can trap it sufficiently with available mosquito traps. So there are studies out there, and we very much appreciate our colleagues from Sudan and Ethiopia, especially, who have been working hard on this. I would like to end by providing some good news here on behalf of my colleagues. We have a new project called ANOSTEP. This is led by Professor Samson Kiwari at Ifakara Health Institute, together with Dr. Eric Uchomo at Kenya Medical Research Institute. It's a large consortium with a number of partners across Africa. We're working in Djibouti, Nigeria, Kenya, Tanzania, Ethiopia. And the idea here is really a comprehensive assessment of the biology and the public health importance of Anopheles stephensi in Africa. We hope that at the end of this 5-year grant, we will have very good evidence of what is happening here. Where is it? What's the best way to trap it? How do we mount proper surveillance techniques here? And what can be done about it? And how, is it really contributing? So I would like to end there. We could have a brief discussion later about that, but thank you all so much for the invitation. Thanks to my colleagues also working on stephensi with us, and back to you, Claire.
Yeah, Fredros, you allowed us a peek into the big bin of challenge that Anopheles stephensi confronts us with. Fascinating, very important presentation, also very well presented. Thank you for that. You raised many questions, as also many concerns. The fact that stephensi shows high levels of resistance to the suite of insecticides that we tend to use to combat our vectors. And you touched on larval source management and so on. Lots of questions. I found fascinating your statement about the use of spatial emanators in buses to prevent the spread, cross-border spread, for example, and wider distribution of the mosquito between cities and between countries and so on. Excellent idea, that. So lots of questions and issues. Thank you for introducing us to those issues, Fredros. For the moment, I'm going to move on to our next speaker, who is Professor Ng Li Ching, with a highly relevant presentation on dengue on the rise, addressing a growing threat. So Associate Professor Ng Li Ching is the Group Director of the Environmental Health Institute at the National Environmental Agency in Singapore. She's also Director of the World Health Organization Collaborating Center for Reference and Research of Arboviruses and their associated vectors. She has spent more than 20 years building scientific capabilities for Singapore's environmental public health, conducting research to understand disease risk and transmission, and developing tools and strategies for mitigation of these risks. She holds a Doctor of Philosophy in Microbiology and a Bachelor of Science with honors from the National University of Singapore. So clearly we have a highly professional and accomplished person to share insights and wisdom, and we look forward to learning from you. Over to you, Li Ching, please.
Thanks, Dr. Liu. Can you see my slide now?
If you wouldn't mind going full screen, please,
Yes.
Professor Ng.
Okay, let's see. All right, it's happening again.
Take your time, no
All right.
panic, take your time.
Okay,
There we
thank
go, you
you.
got it. Thank you.
Yeah, all right. Thank you. I think it's so the flow is really quite good from malaria to dengue and in fact urban malaria to urban dengue. So dengue is very prevalent, it's on the rise, especially because of urbanization. Aedes mosquito, Aedes aegypti especially, which is a very efficient vector for dengue, is an urban dweller. They only feed the females that bites only feed on human. So they really just follow human being wherever we are. And whatever that we have built for ourselves in our urban setting, they have taken advantage. So this is a very clever mosquito. Why go out to the forest under rain and shine? Just go to the home that people have built, right? And stay with them. You get your meals, you get your nursery, you can breed in these homes, you rest in these homes. So very clever mosquito that utilize our facilities or infrastructures that we have built for ourselves, including all these artificial containers. So Aedes aegypti, just very much like Anopheles stephensi, they like urban domestic containers, artificial containers that human beings have developed and put around for our own use, and they take advantage of it. So all these urbanization, I think in the last maybe 10 years, more and more, the number of people living in urban setting has surpassed rural setting. So as more people go into urban setting, we are actually driving the spread and the proliferation of Aedes aegypti, a very efficient vector. A secondary vector of dengue is in fact, it's not moving. Okay, let's see. It's Aedes albopictus. Sorry, I think it's popping up. I don't know why it was delayed.
Yeah, Professor Ng.
Yeah.
Okay, sorry. Yeah, I was just going to query whether you're moving ahead with slides because it wasn't moving
Yeah.
from our perspective, but it looks like the slides are now moving. Thank you. Please proceed.
So another key factor that will be driving, that has been and will continue to drive dengue, is climate change. In fact, this is very well demonstrated in the situation in Nepal. I like to use Nepal because of the terrain of Nepal. You have from more or less sea level and all the way to a very high altitude that represent, in a very short range, representing the different climate from tropical to even Arctic kind of a climate with glacier. So in a situation like that, and with very nice data collection from Nepal, you can actually see how dengue and Aedes aegypti, following Aedes aegypti spread, has been spreading across these mountains from low hills like Terra from 3,300 meters, and it's in more recent years has been found in 2,100 meters with very cool weather. And this really demonstrates the impact of climate change, where winters are getting shorter and warmer, and summers are longer. So all these are driving the invasion and the spread of Aedes aegypti and dengue. So today, I always like to say that this is in fact like dengue and Aedes aegypti climbing mountains, climbing hills in Nepal. This is not unique to Nepal, it's just demonstrating how the adaptation, because of climate change, these mosquitoes are really adapting to new areas, as we have seen all over the world. Globally, about half the world's population are at risk of dengue. 104 to 400 million infections are occurring every year, according to WHO. So in Singapore, we have been suffering for more than 5 decades of dengue. Very similar to Southeast Asia, we all have we're hyper-endemic with dengue, with 4 serotypes circulating in the last 5 to 6 decades, as far as we know. So in Singapore, we have developed an integrated management system for dengue that focus a lot on environmental management and biological control, with only judicious use of chemicals. So some of the principles that we have adopted is, number one, inter-epidemic surveillance and control. dengue comes and go. Every year, every few years, you get a big outbreak. Otherwise, you get this endemicity, low-level endemicity, and then you get an outbreak that often in this part of the world, in Southeast Asia, could overwhelm health systems. So we have developed a system where we say, how can we predict outbreak? How can we work during inter-epidemic period? We don't try not to just react to an outbreak, but work between outbreaks to moderate the incidence of dengue. So it is a whole year-round preventive action with integrated surveillance to guide decision making. The second principle is actually risk-based prevention and intervention. No matter how much resources a country has, it's never enough. So it is very important to do risk assessment, where the important area, where the hotspot, when do you, in fact, in space and in time, when do you pick up your response, or when do you tone down? So these are all important aspect of dengue control and very much driven by data. Very important is the whole of government approach and very intersectoral collaboration with people and with the government, and in fact integrating different fields of science, whether it's field, lab, medical, and epidemiology, and transdisciplinary to solve this problem of dengue. So people, community engagement, community participation is a very important aspect of dengue control. Finally, our final principle is the use of technology and data analytics. It's about making paradigm shift. I will present to you our project Wolbachia and our use of gravity traps. We have been fighting this for many years, and I think it's often not enough to do just more of the same thing, but looking at how different can we do things and how can we increase our productivity as we fight against dengue. So Project Wolbachia is a paradigm shift in our approach in tackling the mounting challenge of dengue control. You can see in this cartoon here, the male mosquito, the male are the one with a bushy antenna. We infect the male mosquito with Wolbachia, which is a natural bacterium. It's natural because it's found in other insects. When you release this mosquito and when this mosquito mates with a female that is in a field that do not carry Wolbachia, you can see that the eggs, the resultant eggs do not hatch. So this is a suppression strategy that Singapore is using. We call it the cytoplasmic incompatibility or insect incompatibility technique, the IIT. And if you just release this mosquito in the long run, you get to suppress the mosquito, wild-type mosquito population in the field, which is the competent vector. There's also another use of replacement strategy where you release both male and female. And because Wolbachia is maternally transmitted, when you release also female mosquito, this female will transfer the Wolbachia to the field population. And in the long run, you replace the whole population with Wolbachia mosquitoes. But the good thing about this Wolbachia is that it blocks dengue transmission, or maybe Zika and chikungunya as well. So the concept here is that it is okay to have mosquitoes, but you have mosquitoes that do not spread dengue or Zika very well. It is partial blocking, it's not 100%, but in Yogyakarta, Indonesia, they have shown more than 70% reduction in dengue with this replacement approach. So two approaches, two very nice use of Wolbachia. In Singapore, we are using the cytoplasmic incompatibility approach, and I will show you some of the data that we have collected in the last few years. Today, almost 6,000 households are covered by Project Wolbachia, and we hope to cover 50% of Singapore by end of next year. So this is these are the containers that we have with the mosquitoes in there. We produce them large scale in the laboratory. In our facility now, we produce about 5 million mosquitoes per week. Put them into these containers, and we go down to these high-rise buildings. This is a typical Singapore building where we release the mosquitoes on the ground floor and on the upper floors as well. And these are some of the areas that are now covered by Project Wolbachia. The whole evaluation of the project is performed by this gravity traps that is also in-house developed. We have distributed more than 70,000 gravity traps all over Singapore to serve as a Aedes mosquito surveillance system to help us focus on the area that needs attention. On top of that, it also helps us to evaluate new tools such as the Wolbachia technology. This is just a chart to just show the impact of the technology. These are 2 towns in Singapore with each of them with more than 60,000 homes, high-rise buildings. And as the blue line shows where Project Wolbachia is, and you can see the eraser effect. As we expand the blue lines, the area of release, we see elimination of the Aedes aegypti population. We can suppress it to very low level. And in fact, and this is another trial that we saw more than between 80 to 90% reduction in Aedes population in these areas. In areas without Aedes, without the Wolbachia mosquitoes, you can see the chart on the right side, the right bottom, they remain very red, showing that the Aedes population still remains very high. When we look at cases and risk of dengue, we have seen 70% reduction in dengue risk in areas with releases. So it's very encouraging that we finally have a new tool for dengue, not just one tool, but we have the Wolbachia suppression, Wolbachia replacement, and you can also use SIT, just radiation itself. So 3 very promising tools finally for tackling epidemic dengue. This is just a chart showing some of the components that is needed to bring a program like that forward. You firstly make a decision to implement, whether with prioritization, you implement, and then you set objective, you have production and release, and the indicators. At the bottom, very important, is the community engagement and the monitoring system. I will end here and perhaps we can discuss further on all these components if we are interested. All right, thank you.
Professor Lee Ching, wonderful, excellent presentation, wonderfully presented, very, very, very interesting, very engaging. So taking us in a wonderful way from an international perspective, explaining Nepal and bringing in the element of climate change, the impacts that is having, and then narrowing down to the various options available for controlling Aedes aegypti, albopictus, and arboviruses, replacement, and so on, and using Singapore statistics to show the impact of those interventions. Well done, very interesting and well presented, and we thank you for sharing your time and experience with us. We're still going to come back to you with questions during Q&A, but in anticipation or in advance already, very sincere thank you for contributing your expertise during this webinar. I'm going to move on to our final presentation, which is by Dr. Tim Harvey-Samuel, who will share insights on the topic of exploring genetic tools for the control of avian malaria in Culex quinquefasciatus mosquitoes. As impactful as human malaria is to many millions of people in the world, so too avian malaria is of major importance in the health of many birds. And we are going to discover more about the fascinating work that Tim and his colleagues are engaged in. Dr. Tim Harvey-Samuel is a lecturer in anthropogenetics at Keele University, specializing in innovative genetic control strategies for insect pests affecting health, agriculture, and biodiversity. He holds degrees from Imperial College London and completed his PhD at the University of Oxford. His research spans gene-drive technologies, molecular biology, and insect sex determination, with previous roles at Cornell University, Virginia Tech, and the Pirbright Institute. Very well qualified man he is. He currently leads a team of researchers exploring the potential use of gene-drive technologies as a tool to control Culex quinquefasciatus mosquitoes, the species which has played a tragic role in the decline of native forest bird populations in the Hawaiian Islands through the transmission of avian malaria. We look forward to learning from you, Tim. Really look forward to your presentation. A little bit of a different angle to what we normally hear about malaria, but in many respects, equally important if we look at the bigger picture of biodiversity in general. So over to you, Tim.
Great. Well, thank you very much. Let me just share my screen. Can you guys see my screen now? I'm in presenter mode now. Oh, it's
there we go. Excellent. Thank you, Tim. Please go ahead.
Cool. Yeah, well, first of all, thanks you very much for the opportunity to speak to you today about some of the work we've been doing in my lab, trying to translate some of the genetic tools that have been developed for the control of human disease vectors, and we've heard some about those today, but now moving them into more of a biodiversity conservation context. And specifically, we're at the moment, we're looking at trying to control avian malaria in the Hawaiian Islands and other islands through developing a genetic biocontrol strategy for a mosquito species, Culex quinquefasciatus. So before I get into the technical details, I thought I would give you a bit of a background because, as Leo said, I'm pretty much the only person speaking about a non-human disease today. So I think that's important to give you the reason why we are working where we are. So when I say Hawaii to you and what most people think of Hawaii, I think they think of something like this. So blue, beautiful blue sky, blue sea, wonderful oceans, beaches. You might have been lucky enough or dreamed of even having your honeymoon there. But when wildlife biologists think of Hawaii, they know it by a different name. They know it as the bird extinction capital of the world. Now, Hawaii, like a lot of remote oceanic islands, has a huge number of what we call endemic species. So these are species that are only found on that island because they evolved there. And what usually happens is at some point in the past, one species has moved to that island, is immigrated to that island, and over time, because there's a niche they can move into, they radiate. They go through these evolutionary radiations, and that is what has happened in Hawaii, especially. This is well known for bird species. So if you think about Darwin's finches, the same thing has happened in Hawaii. And in Hawaii, it is especially well known for its forest bird communities, which are extremely diverse and unique to that island chain. And one of the great examples of that is what are known as these honey creepers. Now, the issue with a lot that it's not only on Hawaii, but a lot of these other Pacific islands, remote islands, is that these unique species have gone through major declines and extinction declines over the last century or two. And we can use the honey creepers here as an example. So pre-Western contact of Hawaii, there were at least 50 species of honey creepers. Currently, we have only 17 species left in Hawaii, so 66% extinction rate over the last 2 centuries. However, even more critically than that, 11 of those that are remaining in Hawaii are considered critically endangered. So really, there's something that there's an immediate situation that needs to be addressed if we are going to retain these unique and very important species that have evolved in these Pacific islands, including Hawaii. And the reason why these bird populations have declined in Hawaii, and this is a situation that's played out in lots of Pacific islands, is through the introduction of invasive species. In Hawaii, it's a little bit unique because normally these invasive species are predators or even herbivores, but in Hawaii, it's actually a vector species. So that's quite unique. So what we know is that prior to Western contact, there were no mosquito species in Hawaii. because of its remote location. However, with Western contact, we know that Culex quinquefasciatus, the first mosquito species, was brought to Hawaii by a whaling vessel in the 1800s. And it rapidly established in Hawaii because Culex are bird-biting mosquitoes. And there were lots of these bird species on Hawaii for them to feed on. However, it wasn't until the 1940s that we started to see a real major decline in these bird populations in Hawaii. And that's because at this point, Plasmodium relictum, which is the causal agent of avian malaria, had become established in Hawaii. So this is quite interesting. It shows you need both the vector and the disease-causing agent in order to cause these die-offs. And the reason why the birds of Hawaii were so affected by avian malaria, which is found all around the world, is because, like a lot of island species, they had no immunity. They had evolved or retained no immunity to Plasmodium relictum. So it really, really badly affected them when they became infected with this novel disease. But the situation, like I said on the previous slide, is even more dire than you would imagine, because if we don't do something, the majority, if not all of these species will probably go extinct in the next century. And the reason for that is because those that are currently still existing in Hawaii are only really existing in what's known as a refugia area. And it's very interesting to follow Professor Ng's presentation about Nepal, because exactly the same situation is happening in Hawaii with Culex. So what we know in Hawaii is that it's a high island. So like Nepal, there are actually, you might not think of this, but there are actually snowy areas in Hawaii as well, because it's so high up these mountains. And on these mountains in the low forested areas, mosquitoes and malaria can exist because it's warm. And these areas, there are no birds left. The birds have all gone extinct. It's a bird exclusion zone. The birds can only really exist in these refugia areas halfway up the slopes, where it's too cold for malaria and Culex to persist. However, what we know and what we are seeing is that actually through global warming, these thermoclines are actually moving up these mountain areas. So these refuges are actually shrinking every year. And these are forest bird species. They need to exist in forested areas. So they're being pushed up further and further against the tree line. And eventually they will run out of room and will go extinct as the mosquitoes and malaria transition up the slope. So this is a really critical and imminent situation. We need to do something now if we're going to preserve these species. The solution that we're looking at in our lab is to develop what's known as a population modification gene drive. So a lot of people will be familiar with this type of technology, but just to go over briefly, the idea with a population modification gene drive is that you are spreading, you're engineering a system in the mosquito species that is able to spread a trait through a population of that mosquito, which makes it less harmful, for example, here to birds. So the trait we're thinking of spreading is a trait which makes the mosquito individuals that inherit this transgene, this engineered gene, unable to vector or transmit Plasmodium relictum. So effectively, it means they are immunized against this carrying this disease. And the gene drive part of this is that we are engineering a system which spreads that gene, that controlled trait through the wild population of mosquitoes for us. And the reason we're looking at this particular type of strategy is because if you look at, unlike, for example, what Professor Ng was talking about in Singapore, the area we would be trying to control Culex in Hawaii is extremely remote, extremely rugged. You can't think of actually applying. It's very difficult to consider traditional vector control in these types of environments because they're just impossible to get into. Even with things like the sterile insect technique or IIT, where you have to constantly release the insects into these environments to suppress the populations, it would be very difficult, very costly to do that over the whole of Hawaii. So we're thinking of developing these systems that can actually spread the anti-malarial genes through the population of mosquitoes for us. So this project consists of 3 components, and I would like to point out at the beginning that this is not something new that we've developed. We've leaned very heavily over a lot of success that has happened over the last few decades, trying to develop such similar systems for the control of human malaria in Anopheline mosquitoes. I think Fredros also mentioned this previously. So two groups in particular, Transmission Zero at Imperial and UCMI at the University of California, have done a lot of great work in developing these gene drive systems to control, to make mosquito populations immune to carrying human malaria. And we've, as I said, we're leaning very heavily or influenced very heavily by that work that has gone before us. So in order to build one of these systems, we need at least three components. We need to develop a system, we need to identify a mechanism to make these mosquitoes, Culex quinquefasciatus, unable to vector or carry the disease-causing agent, in this case, the sporozoites of Plasmodium relictum. In our case, we've gone down an antibody discovery route, so similar to the UCMI approach. You need a set of tools to be able to express these antibodies, the antibody variants within the mosquito species at the right time and at the right place. So we need to identify suitable promoters for that, to engineer these mosquitoes to make them blood meal inducible, for example. And we also need to develop a gene drive system to spread these two components through the mosquito population for us. And while we're research in this is ongoing in all three of these parts, I'm going to be talking about our work in developing the drive system predominantly today. So the type of system that we're developing initially is what's known as a homing gene drive. And for those of you who aren't familiar with this technology, I'll go over it very, very briefly. The idea is, with a homing gene drive, you're engineering a mosquito with a transgene which makes copies of itself within the genome of that species. And ideally, it's doing that in the cells, which will go on to become the next generation. So in the sperm and the eggs of that mosquito species. So let's imagine, first of all, we have a normal gene in a normal transgene in a mosquito. If this mosquito was a heterozygote, it had one copy of that gene, then that gene would be passed on to 50% of its progeny. What happens in a gene drive is we engineer that gene to express at least two components, a targeted nuclease and the targeting component, Cas9 and the guide RNA. And these cut the homologous chromosome within these germline cells. They create a break, and the cell repairs that break, hopefully by transferring or copying the gene drive into the break. So now we say that the wild type chromosome has been converted to carry the gene drive. So we've gone from an individual that starts off as a heterozygote and is now a homozygote. And again, if you think about this at the population level, it creates this spread of the system. Similar to PCR, you're getting this exponential increase over generations in the inheritance of these transgene components. So where are we at in terms of developing this in Culex quinquefasciatus? Well, we have had proof of principle success. We published this in 2023. If you remember on the previous page, a Mendelian inheritance, a normal inheritance of a transgene would be 50%, i.e. to half of the progeny if it in the heterozygote. We achieved over 50% inheritance, known as super Mendelian inheritance. So we actually observed 56% inheritance of the gene drive in individuals which carried the engineered genes. So what that means is that around 13% of these wild type chromosomes had been converted to carrying the gene drive transgene. So this was really exciting because this is the first time that such a system has been engineered in a Culex mosquito, which traditionally have fallen behind in terms of these genetic control techniques. But of course, 56% inheritance is not going to be good enough for a field strain. So we need to optimize this system. And this is what we're working on now, leaning heavily on work that has been done by my colleagues at Kiel, Roberto Galizzi, as well as his collaborators at Liverpool School of Tropical Medicine, Tony Nolan. So we're looking at trying to figure out if we can improve the expression of Cas9 within the part of the germline, the gonads, which we think is really important for this homing reaction, this converting reaction. And that is the pre-meiotic stages of the germline. So in order to do that, we are doing single cell sequencing of the male and hopefully also the female germlines of Culex quinquefasciatus to try and identify genes which are highly expressed in these cells in order to use their promoters and regulatory elements to increase the expression of Cas9 exactly where we need it to create this homing reaction. And I'll leave it there. Thank you very much for listening. Our latest project on single-cell sequencing is funded by the Academy of Medical Sciences, and these are the people who have helped us along the way and continue to collaborate with us on this project. But thank you very much.
Tim, so thank you for explaining so very clearly, logically, the problem that is being faced in Hawaii. It's distressing to hear 66% extinction rate in a particular group of birds, creepers, and the future looks pretty bleak given climate change and the transition up the mountain, creating access for mosquitoes and the parasites, and the birds being limited by the tree line. And please know that I think everyone in the audience will agree. We're keeping our fingers tightly crossed and hope that you can come up with a higher than 56% rate success in the gene drive efficiency. And we wish you very, very well in your research. Excellent presentation, making it clear, not just the context, but exactly the tool that that you're working towards, and the success of it. So well done, and thank you for that, Tim. Just for the benefit of the audience, an interesting little snippet. It seems like we're hovering around 448 participants in the audience, which is a good number of people. It shows that the webinar has attracted a lot of attention, and thank you to all of you for showing such interest and continuing to listen in to all our presentations, which have now come to an end, very unfortunately, but we still move on to another equally important session, which is our questions and answer session. During the 4 presentations, we learned that it's not just the traditional 4 species of so-called human parasites that cause human malaria, those being Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. But there exists a bunch of others in Asia and South America and Africa, that are hidden in monkey or primate populations and will be more difficult to eliminate than human malaria parasites. We learned that malaria parasites are not limited to primates either, whether they be humans or monkeys, but that even birds are plagued by their own malaria parasites. And there are others in rodents, in reptiles, and more. We also learned about the rise in arboviruses, especially dengue, and that across the world, our human future will have to deal with more and more outbreaks, such as we are currently seeing with chikungunya. And we learned about the serious threat facing Africa by way of urban malaria vector, Anopheles stevensii. This introduced species could single-handedly reverse the gains we have made in malaria control in Africa, or at least pose a serious challenge in our attempts to eliminate malaria from that continent that suffers 95% of the annual global malaria burden. So let us tap into the wisdom of our expert panel to extract a few more nuggets from them. I'm going to intersperse some pre-prepared questions to them, and then we mix it with questions from the audience. So here we go. The first prepared question is to Professor Kimberly Fornas. Kimberly, you've already touched on the aspect of vectors, just touched the surface over there. We know the vector species of mosquitoes that transmit the parasites of the human malaria parasites. And we know their behaviors, so we can try and target interventions to control them. But what about the mosquitoes that transmit simian malaria? Do we know which species are responsible for transmission and the behavior? You mentioned Anopheles leucospirus. Would you mind just elaborating just a little bit, just briefly? What is the overlap between the traditional human-biting, anthropophilic vector species and the more zoophilic species? And, any thoughts on how do we deal with what you seem to suggest of vectors that are biting early evening before people go indoors and, sleep under their bed nets where they will be protected? So any thoughts from you on that, Kimberly?
Yeah, it's a really interesting question. I think there's certainly an increase in the amount of data on the different vectors for Plasmodium knowlesi and other kind of zoonotic or simian malarias within Southeast Asia. So in some cases, we find this is the exact same vector as was primarily transmitting non-zoonotic malaria. So for example, in Sabo, Anopheles balabacensis was the main vector for non-zoonotic malaria. And we think this is the main vector in many parts of Sabah for Plasmodium knowlesi as well. One thing that we've been really interested in, which we have still kind of real gaps around, is not just what's spreading malaria from monkeys to people, but actually what's driving transmission between monkeys. So thinking about at canopy level, what's really driving these sylvatic cycles. And I think that's still really a gap. That's obviously a lot of challenges in trying to think about how to design those studies and how to catch them. So certainly that's an area we've been thinking a lot about and hoping to work towards. And then in terms of your kind of question about vector control, I think it's challenging. There's specific risk groups. If we know that it's outdoor biting, we could think about targeting different types of repellents or vector control to these groups, and particularly during certain activities in specific types of environments. So example, different types of plantations or logging camps where we could make sure to really focus on trying to target preventative measures. So again, yeah, still very much a work in progress.
Yeah. No, thank you for that, Kimberly. That's it's a whole big aspect in itself to deal with. And hopefully in the audience there are a couple of potential MSc or PhD students who would be interested in tackling some of those aspects, such as canopy frequenting vector species that transmit the mosquitoes between the monkeys. Interesting question that. So good luck and thank you for the work that you're doing on that. You're a lady of multiple talents, equally an expert on simian monkey malaria, simian parasites, and also climate change and so on. So I take my hat off to you. We have a whole team of people working in the background to ensure that our Zoom platform is functioning optimally and coming up with screen displays and so on. One of them is Joyce, who has been monitoring the questions coming up in the Q&A box. And because unfortunately, we won't be able to deal with all the questions that come up. We have 15 minutes left of our allocated 90-minute webinar. So in those 15 minutes, we're going to alternate between some questions from the audience and then a few more questions pre-prepared for our panelists. So Joyce, we ask that you select one or two or a few of the questions from the audience that may have broad appeal for a wide range of audience members. Here's the first one that Joyce has kindly selected for us. Kimberly, given the dual challenges of climate change and biodiversity loss, how can integrated vector surveillance systems be designed to simultaneously protect human health from mosquito-borne diseases while also safeguarding ecosystems and species that are equally vulnerable to these changing environments? So let's give a couple of seconds for Kimberly to wrap her mind around this question. Kimberly, when you're ready, do you want to have a go at this question, please?
Yeah, it's a great question. And just seeing the other presentations, I imagine it's something the other panelists would also have quite a lot to say about, because there's certainly a lot of common themes that are coming out across these different presentations and different species of vectors or viruses as well as parasites. So we've been really thinking a lot about, rather than just thinking about kind of land use change as a main driver, but also thinking about how we design more resilient landscapes, and ideally designing landscapes where we can try to support biodiversity or carbon goals as well as mosquito-borne diseases. So I know certainly within Malaysia and many other countries, we're looking at reforestation. So trying to understand the impacts of reforestation and actually thinking about co-benefits. So, specific types of practices that might benefit these different goals for kind of biodiversity and ecosystem services, as well as actually kind of reducing malaria risks or reducing vector populations. And certainly that raises a really key question about how to monitor multiple outcomes and how to really integrate vector control, looking across kind of different species or different outcomes that are of concern as well.
Thank you, Kimberly. Thanks for that. I hope that adequately answers the question for the person who posed that question. Thank you for selecting that very interesting one, Joyce. Thank you for that. So I'm going to jump now to Fredros. And the pre-prepared question, Fredros, is that Anopheles stevensii poses potentially a game-changing threat that risks us seeing an increase in annual malaria cases in Africa and in future at continental scale, such as you showed in your graph for Djibouti. From where I sit, my little ignorant individual self, my impression is that the malaria world is pretty much treading water, encouraging countries to strengthening their surveillance and to do yet more research. But I don't see any incisive recommendations to stop the rot and reverse the invasion. I know that there's a lot of work going on gene drive, on Anopheles stevensii, but this could take some years yet, almost certainly. What are your thoughts on how we should proceed with hopefully more incisive actions? Over to you, Fredros.
Thank you very much, Leo. So the short answer is I don't know, but let me see what I try.
Thank you for being honest.
I sympathize with this position that Africa should aggressively go to war against Anopheles stevensii. I wonder, though, whether, number one, Anopheles stevensii is the only invasive, dangerous organism in Africa. I wonder what the people who say this would say about Anopheles Aedes albopictus, for example, which is also spreading in Africa. And number two, I wonder whether the amount of resources or capabilities that would be required to go to war against Anopheles stevensii would be justifiable given the proportion of malaria that Anopheles stevensii currently contributes vis-a-vis the proportion of malaria contributed by the big four: Anopheles gambiae, Anopheles funestus, rabiensis, Anopheles columnsii. And so someone might say, well, if we didn't go to war aggressively against Anopheles gambiae or rabiensis, what is the justification for stevensii? Having said that, however, you remember during my presentation, I said that, people who live in urban Africa have more power than people who live in rural Africa. If you have a single case or two transmitted by Anopheles stevensii in one of these urban cities, it will go to the newspapers, it will go to New York Times, it will go everywhere. And these guys will start demanding control. And so the NMCP might say, okay, well, political reasons, we're going to distribute bed nets in Dar es Salaam, or we're going to distribute bed nets in Nairobi. And what is that going to do to the bed net portfolio? And how much will be left to deal with malaria in the rural areas? And so on one hand, I sympathize with the idea that if we do not address it aggressively now, it might spread and become something super big in future. Maybe. Probability is not zero. But to balance things, I think that we should take a more integrated approach here. We should apportion 80% of our resources to dealing with the big deal, the big things, and then 20% of the resources to dealing with an awfully stiff insect. That 20% of the resources will not be sufficient for a militaristic approach against the vectors. So this is why we've got to be long-term thinking and integrated. Having said that, I do know that there are some new approaches that are being developed that might actually be low cost and that might actually be scalable enough to go to war with an awfully stiff insect. Case in point is the work that Oxitec is doing in Djibouti and the work that is being done around genetically modified mosquitoes. You know, one of the concerns about genetically modified mosquitoes is from biodiversity groups that say, oh, what happens if you eliminate species X? Why is it? Those concerns will be less if it's an invasive species. We might be more justified to go to war with gene drives against typhus. And that will be low cost, very cheap, and species specific. And therefore, if we could develop that, it might be a path. But if we're going to go to war against typhus with IRS, ITNs, larviciding, I will ask, why don't you do the same for Gambia, Arabiensis, and all that? So this is, you can tell from my speech myself that I'm not sure if my answer really makes sense. But whereas I think I sympathize with that position, I don't think that it's appropriate for low-income settings under the existing financial settings. Leo, is that helpful?
Well, it's food for thought, Fredros, and certainly, it made me realize there's at least one webinar and possibly 2, 3, 4, 5 webinars around this particular topic of do we sit back and just see how the Stevens Eye situation unfolds and say no, available resources should be devoted to the existing known priority of Gambi, Funestus, Arabiensis, and so on, for which we have tools and we have populations that have developed a certain level of immunological tolerance or resistance, whereas Stevens Eye as an urban vector represents a whole new paradigm shift, kind of a naive environment where very little control efforts are being directed and immunologically naive population, highly susceptible to infection as well. So it reminds me of, we both know him, Bart Knowles. He always likes to hold up, and I'm sure you will know about it. He likes to compare the situation with Fred Soper. If we could eradicate Anopheles gambiae in South America through concerted dedication, why can't we do the same for Stevens Eye? But let's not dwell on that because we have another 5 minutes left. So Fredros, thank you for those thought-provoking statements. Definitely.
Leo, real quick, if that war was mounted, I would be on the front lines. Yeah.
Okay.
So just so you know.
All right.
But only if the tools are appropriate and we can justify the resources. You understand?
Yes, and of course the resources have to be made available, and they're not available right now. Yeah. No, thank you very much, Fredros. So I'm going to move on quickly now because we're running out of time. Professor Lee Ching-Eng, your country, Singapore, has made highly impressive interventions against dengue with great success. And other examples exist in Asia as well. But I'd like to extend my congratulations to you and your team. Singapore has done extremely well. Impressive work that's going on there. Vaccines are probably the future for widespread control, but for the moment, there are obstacles to widespread adoption. Wolbachia has proven to be a very effective intervention, as shown in your own country, also in Indonesia and elsewhere. Other countries are exploring adopting this approach. What do you say to these countries in terms of what they need if they want to adopt such a Wolbachia approach? I'm thinking about unrealistic expectations, about the scale of impact, funding, local entomological expertise, social readiness, and so on. Any thoughts from you, please, Professor Ng?
Yes, in fact, the gears that I showed just now, the different gears, it's
Yes.
really all the different components that are very much needed. They work like an engine. If you have one missing gear, the engine can't move, right? So every single component is important. We start from the decision making. We mentioned about decision making. So over there, we've done a lot of risk assessment, getting laboratory data to support, to get data, to get evidence that this could really work in the field. So that decision making, and of course the funding as well, a commitment to say, if this work, I'm going to expand it, I'm going to scale it up. So I think that component is important. And then you have the public engagement, community acceptance. I think there's a question related to that. It's very important because you're going to release mosquito into the field. So some of the things that we have done, again, you show data, you show risk assessment, and you give assurance that there's no risk. Science communication is important here. Often when we speak science, it's difficult to understand, especially if you talk about what female mosquito, cytoplasmic incompatibility and all that. So how do you then shape your communication content to what and your audience would like to receive, how they would like to receive it. So science communication is something that we spend a lot of time on, engaging the public, engaging experts, engaging stakeholders to understand their concerns, and then we address these concerns. So that is very important as well. The other component is actually scaling up. How do you scale? So this is not a turnkey technology. We couldn't buy any equipment. I want to count the number of eggs that we put it in, or larvae. How do you count them? There was no equipment to do that. So we had to develop everything from scratch. So today they are available. You're welcome to visit our facility to see them. But when we started, they were not available. So we have to get engineers, biotechnologists, and all these come together. So overall, I would say, and of course, then you have the evaluation tools. You need the evaluation, the data analysis. So overall, I would say the whole thing, it's not a single laboratory, it's not a single agency that works. It is a collective effort ranging from biotechnologists, social science, engineering, data analysts, all put together to make this work. So I'm very appreciative of all our collaborators out there that has come along, taken this journey with us together. Yeah, thank you.
Thank you so much, Professor Li Ching. And again, from all of us, sincere, deep congratulations on the work that you're doing. They're a wonderful, admirable example of how to do things and make it work. Just to mention, we're still over 400 people in the audience, but you can see interest flagging. People are dropping off pretty steadily. So I'm going to make this the last series of questions. I'd like to ask audience members right at the end, Wulan is going to, or somebody is going to pop up on the screen, a very short, Dr. Porn is going to put on the screen 3 questions, multiple choice set of questions. Then you just need to tick the box. It will take you less than a minute. Just a short survey to tell us, the organizers, what we can improve on, what we did okay with. It helps us. So, Joyce, I'm not sure if you were able to select a question for Professor Ng from the audience Q&A. Okay, so there it is, Li Ching. What public perception or acceptance challenges have you encountered in rolling out the SIT or Bahia program in Singapore, and how have you addressed them? SIT is a sterile insect technique. How have you addressed these issues? Please, your insights, Professor.
Yeah, because it is so new, so there's a lot of unknown to the public. Of course, we have a lot of data to give us the assurance, but the public didn't have the data. So it's very important for us to demonstrate to assure them. And one very important, it's very difficult concept, right? That it's ironical that you release more mosquitoes to reduce mosquito. How do you wrap your mind around?
Counterintuitive.
Yeah,
Good point.
counterintuitive. So that's something that we have to. In fact, in Chinese terminology, in our local, one of the local languages, there is a term. You use poison against poison. So it's sometimes you use local terminology to help you explain. But one very important difference is that the males. Very important for us is that the males don't bite. The ones that we release don't bite, they buzz around you and all that. So that is a very key message for us. We know it works. You may get irritated, you may get buzzing mosquitoes, but they don't bite. And so some of this very key information, we need to push it out to give people the assurance. And then we use data as to be very transparent with our data. Whenever we get data, we surface it to the public and say, is it working or is it not working? So the first trial that we had, a very small one, it didn't work. And we had to redo and rethink our experiment, rethink the study. And we came up very outrightly to tell the public, sorry, this time we didn't work but we have learned all these lessons and we're going to try again. So that I think it's very important to be transparent. And I mentioned about the language used and key information that is very assuring. We put it out. So when we started, I think people were saying, why me? Why my area? Why are you coming to my area? Why do you select my area? So we had to put up data to show them why your area, because you used to have a lot of dengue. But I think today we are getting questioned like, why not me?
Yeah.
So we
Very
have
good.
96% of the population that we surveyed, multiple surveys showing 96% either have no opinion or accept the technology. Yeah.
Thank you for those insights, Professor Ng. Much appreciated. So, Tim, you next. Many see gene drives as a promising tool for malaria and vector control. Looking ahead, what do you see as the most challenging areas to address to be able to successfully develop gene drive systems to control disease-carrying Culex mosquitoes, or any of the other vector species for that matter? But using your Culex quinque fasciatus maybe as a model. Over to you, Tim.
Yeah, I'll keep it broad because I'm aware that most people on this webinar are interested in human diseases. But that also includes Culex as well. So I think it's important to note that there's been a huge amount of development in gene drives, but in the vast majority of species that they've been tried in, these homing drives, they haven't worked very well at all. Now, Anopheles mosquitoes, for some odd reason that no one really knows, work extremely well. So if you remember the homing rates or the conversion rates I was talking about before, we on our first attempt got 56% inheritance. Pretty much the first attempts that they did in Anopheles, at least the ones they published, were over 99, so are close to 99. So it's a completely different story. So it really depends. Your question really depends on the answer to your question depends on what species you're going for. Anopheline mosquitoes right now, as I pointed out, and a lot of people will be aware of this, they have strains right now that are, theoretically ready for field testing. They're that good. For the other mosquito species, Aedes mosquitoes, specifically Aedes aegypti, there's been a lot of work on trying to improve those rates, Culex as well, and it can possibly be done, but it seems to be much more difficult. So yeah, basically from a technical point of view, you need to be able to make them much better at spreading. And also a separate issue is you need to make them robust enough to be able that they don't generate resistance against themselves as they spread. And there's ongoing work on both of those points. For example, in Aedes aegypti, we're at a pretty early stage in Culex. There's just taking Anopheles as another example in terms of the resistance management. There are some tricks that people have come up with to be able to be fairly robust, we think, against resistant development in those as well. So really, Anopheles is leading the charge here in terms of gene drive development. Does that answer your question?
Yes, it gives us kind of a general idea. So thank you for those thoughts, Tim. And we're rapidly losing participants. We're still at very good numbers, but the numbers are dropping, reach the end of attention span, and also time allotment. So I'm going to ask that we forego on remaining audience questions, but let me reassure the audience that we will harvest all your questions and we will pose them by email to our wonderful panel of experts and ask if they don't mind, if they could address those questions, return them to us, in which case we will then post them so that your questions will be answered. Please be patient, give us a few days. Together with the presentations, we hope to have the questions posted to you as well. I'm going to ask Dr. Porn and or Wulan or whoever, please just, yes, thank you for that. Here's the satisfaction survey. Please, if our audience members, if you can just respond to those questions, tick the appropriate response. Let me take a moment to thank each of our distinguished panel of speakers for your time, for setting aside valuable time, of which none of us have enough of, but to participate in this webinar, share your experience and insights. It's been a wonderful, genuinely great webinar made possible by you. To our audience, thank you for your interest. Thank you for hanging in there despite us going over time. You've been a wonderful audience, and we thank you for your interest. And as we say, we will make the entire webinar, which has been recorded, available to you, and also hopefully also all the questions hopefully will be responded to and uploaded as well. So to everyone involved, and also not to forget our technical team, our background team, Dr. Pond-Willan, Joyce Tamson, everyone else, a sincere thank you for enabling this webinar to take place by ensuring that the platform and the technicalities are dealt with. So deep and sincere thank you to all of you. Go thee well. Be good. Thank you.